patterns, like these striking warning markings of hoverflies, wasps and bees, the cryptic coloration of moths, camouflaging them against their bird predators, and the markings of these animals are all familiar everyday examples of the complex patterns which are generated during development. We're less familiar with the patterns produced during early development. Cells divide in a blastula and take up specific positions. They form a simple pattern, yet this pattern will determine the final form of the developing embryo. But what are the stimuli which tell cells to produce patterns? And how do cells respond to their developmental instructions? During neurulation, cells migrate to form a nerve cord, and the outline of the embryo emerges. So cell migration is certainly one response to developmental instructions. But other responses, such as differentiation, may be brought about by biochemical or other gradients. To attempt to explain complex developmental patterns is too difficult. We need first to look at simple systems, and these may provide us with model answers. Growing in this lake is the blue-green alga Anabina. This simple organism exhibits one of the simplest of developmental patterns. Single filaments of Anabina consist of two types of cells: vegetative cells, and these more circular cells with thicker walls, the heterocysts. Their function is concerned with fixing atmospheric nitrogen, and their differentiation is geared to producing an oxygen-free environment in which an oxygen-sensitive enzyme catalyzes this fixation. The pattern we're interested in is the regular spacing between heterocysts. Heterocysts can't divide, and when the filaments grow, the number of vegetative cells between adjacent heterocysts increases, and then new cells differentiate to form heterocysts, so maintaining the precise pattern. The question is, how is this pattern maintained? Suppose we take a chain of 8 cells and a heterocyst at each end. We could draw a curve representing the concentration gradient of inhibitor. Now, if we take this line to represent the threshold level below which a heterocyst will develop, then all 8 cells are subjected to too high a level of inhibitor and no new heterocysts form. But what happens when cells divide? The interval between heterocysts widens so that the concentration of inhibitor drops until it eventually falls below the threshold in one of the cells. But how with this simple mechanism does only one cell differentiate rather than 2 or a group? Well, Anabina has 2 ways of overcoming this problem. First, cell division is asymmetric. Each cell divides to give a short and a long cell, but heterocysts only develop from short cells. And secondly, a competitive mechanism exists. If 2 cells close together both start to differentiate, then they'll both produce inhibitor, and each is likely to be inhibited by the other. A competition exists for the one available slot. Eventually, one cell produces more inhibitor than the other, and this develops into a heterocyst. The other cell reverts to a normal cell. As the new heterocyst produced more inhibitor, the concentration level increases above the threshold point, and so a stable pattern is maintained. In more complex organisms, the problems of development are more difficult to unravel. What experimental techniques are available to deal with them? This is polyspondylium. It's a slime mold which lives at very low light intensities, so it's difficult to film. Professor Ted Cox of Princeton University has been looking at its spacing pattern.
Polyspondylum, when it's completed its growth phase, has a single long stalk, a ball at the top, but on the way up, it sends out a series of arms. It has whorls of stalk cells that stick out from the side of the central stalk, and these stalk arms terminate in balls of spores. Now, if you look at this, you'll see that it has some resemblances to Anabina. That is, there's an interval here, which sometimes we refer to as the interval n, sometimes we refer to it as I, and sometimes we refer to it as j. The question is this interval controlled or not? Is this distance tightly controlled? And the answer seems to be that it is. It's not as tightly controlled as the spacing pattern in Anabina, but if one simply plots over thousands of plants this distance between adjacent whorls, we get a very nice distribution like this, and it centers about some mean. This mean is not absolutely indeterminate. In heterocyst formation in Anabina, you'll recall that the heterocyst is spaced rather regularly. But nonetheless, we can say with assurance that there is a mean value here, and it's reproducible as long as we control the various conditions under which the organism grows. The next and obvious question is, how does it determine this spacing, now that we've established that it is spaced regularly? One can think of many mechanisms. The two obvious ones are that this organism counts the number of cells along the central stalk. The other obvious one is that it measures distance. And I think one can distinguish between those two very easily, either the counting mechanism or the distance mechanism, by using genetic tricks. Our genetic trick was to make diploid stalks of Polyspondylium, and in Polyspondylium, as in Dictyostelium, the diploid cells are about twice the size of haploid. We then made the diploid cells, measured the distribution about the mean here, that is, how many between the interval, and we found that the distribution was almost exactly the same. Now recall that in a diploid, the cells are roughly twice as big as a haploid. A diploid cell looks something like this, and the same interval would have normally 2 haploid cells. And yet, we cannot distinguish the interval spacing in a diploid from that in a haploid. And that tells us, I think, that the organism, this little plant, measures distance, that it doesn't count number of cells. Now that's one of the interesting questions. Of course, this doesn't tell us anything about mechanism. We don't know whether chemicals diffuse down the length of the animal or the plant. I think it depends whether you're a botanist or zoologist. We don't know whether it's a gas. We might even imagine that it's a small biochemical clock running that simply triggers off events like a ticker tape. But we can now approach this using time-lapse cinematography and using the scanning electron microscope to define these events a little more accurately. One of the things that we've noticed is that when the mass of cells is growing up and ascending, a wave sweeps down the length of this slug so that a little bit later, a few minutes later, there's an obvious bulging out at the bottom, and a few minutes later yet, there's a donut of cells left behind on the stalk, and it's this donut that differentiates into the radial arms, but it's the number of cells in this mass that determine the number of arms that will finally form. The second interesting point is that sometime at about this stage, before the donut of cells is left behind, small organizing tips can be seen forming around the perimeter. They first show as refractal changes, and then later they appear as tips, and those tips then turn into arms, and the arms differentiate further into the stalk cells plus the spores at the end. Now, one basic question that we now have to address with this system is, how do those organizing tips form? Do they form symmetrically? Do they first appear asymmetrically? Does one then dominate the other and extinguish adjacent ones until the right number of tips are there for the right number of arms? That's one question that we hope to answer using mutants that we've isolated, and those mutants have problems with their spacing. Some of them space aperiodically, that is, there's arms growing out everywhere. Some of them space with a short period, some with a long period. We hope by using those mutants now to address this question. Now, one of the interesting features of this question, of this particular problem, is that you can imagine this as a torus of cells, and you can also imagine that there's a wave of diffusing chemicals around the perimeter. There has to be, according to some models, two different waves of chemicals, one extinguishing the other, and one can imagine that these particular chemicals represent nodes of concentration about this torus of cells, and it's those nodes that tell the arms to grow out at these particular points. The basic problem then is how to get at the chemistry. We hope to get at it through the genetics. Why is the spacing here so regular when these arms finally form? And then what is it that keeps the arms regular as they grow out even further away from the torus of cells? A little bit later in this sequence, there's a large pod of cells growing out from the middle on its way to differentiating to stalk plus spores. And some of our time-lapse films show us that these little pods move around in space, and when they get close together, they immediately go apart again. And that suggests to us there might even be a gas involved in the terminal spacing of these arms. It seems very unlikely, however, that anything but a diffusing chemical reaction of some kind will explain the early appearance of these little organizing points around the perimeter of this torus of cells. One could liken this whole thing to a donut machine in which the growing tip of cells leaves donuts behind it and leaves them at the correct interval. Polyspondylum is a little more complicated than at least I first thought. There's a one-dimensional pattern along the length of the stalk, that is, the spacing of the whorls. There's a two- or three-dimensional pattern along the perimeter of the cells that are left behind that will eventually form a whorl. And then from the time-lapse cinematography, we've shown that there's something that spaces the arms out so that they, too, are spaced evenly around the perimeter of the stalk itself. It's not going to be easy to answer any of these questions. We will find it hard, I believe, to get to the biochemical basis of pattern formation in this little plant. But I think that we have genetics going for us. We can isolate mutants that alter spacing, and we can isolate mutants that turn on different biochemical pathways, and hope, by combining some simple biochemistry with a mutational analysis, to settle at least one of the bases for pattern formation in this little plant.
This is a fruit fly, Drosophila, emerging from its pupal case. During pupation, larval tissues change into adult tissues. Most adult structures are derived from a specialized group of cells, the imaginal discs, and it's these that provide us with a higher order problem in pattern formation. Even under higher magnification, it's difficult to visualize what any one disc will produce. This is a larval Drosophila prior to pupation. Imaginal discs can be dissected out from the larva. But unless one can determine what each individual disc will produce, further analysis of pattern formations impossible. However, using metamorphosis itself as an analytical tool, each disc has been identified. When individual discs are isolated, they can be injected into the abdomen of a host larva. During metamorphosis, the discs develop into the structure they would have formed in the intact adult. The metamorphosed disc can then be removed from its host and examined. But because the disc has been floating in the abdomen of its host and not attached to the fly in any way, it fails to evert. It doesn't blow itself out and remains like this a compacted, rather crumpled structure. But under a microscope, however, it's easy to see that this disc is a wing. Other discs develop into different structures. Analytical techniques like this are extremely delicate and difficult to perform, but by using them, we're now in a position of knowing what each disc will produce in the adult fly. But that's not all you can do. Dr. Mary Barnes from Edinburgh University is a leading authority on Drosophila development. And by cutting individual discs, she and other workers have been able to gain a greater understanding of disc development. If a disc is cut, each of the two parts can be implanted into a larva. After metamorphosis, the new structures examined. By making a series of cuts, implanting the fragments, noting their post-metamorphic appearance, a complete fate map of each part of each disc can be made. The different parts of the adult head can be assigned to specific regions of the disc by observing what structures are missing in the head after cutting out part of the disc. Remove this region, and the arista part of the antenna in the adult is missing. So this part of the disc develops into the arista of the adult head. Remove this part of the disc, and the vibrissae, these hairs above the palps, are missing. So this part of the disc is concerned with vibrissa development of the adult head. And by making a whole series of cuts, different parts of the adult head can be mapped to the disc. As a result of analysis by this technique, an accurate fate map of every imaginal disc can be produced. Normally then, fragments of discs develop into their regional specific structures. But if fragments are given more time to develop, by first implanting into an adult and then into a pupa where metamorphosis can take place, two new phenomena emerge. This head disc has duplicated itself down this line. Where normally it would have produced one antenna, it now produces two. This disc has regenerated the missing section, so depending on where the disc is cut, it can either duplicate or regenerate. It's easier to see what happens like this. This is an eye antennal disc, and a cut is made here. After incubation and metamorphosis, the antennal parts duplicate, and the eye part regenerates an antennal region. But how can duplication and regeneration be explained? We can think of the disc as being made up of cells which are in a gradient of developmental capacity. Cells high on the gradient have a higher developmental capacity than cells lower in the gradient, so that cells taken from the eye region develop into antennal structures. Cells taken from the antennal region could not develop into eye structures. So that when a disc is cut here, the cells in the eye part can regenerate the antennal region, which is lower in developmental capacity, while the antennal region can only duplicate itself, being unable to regenerate the eye region, which has a higher developmental capacity. But this model can't explain all experimental observations. So another way of looking at this is by the use of the clock model. In this case, the disc is represented as a clock face. The numbers on the clock face represent positional values on the disc. But there is no greater discontinuity between, say, 12 and 1, or 1 and 2, than between any other two neighboring numbers. In other words, there are no gradients in developmental capacity. That's the model. Now, we made our cut on the disc about here. This gives two separate pieces. These heal, but the healing processes bring together cells which are usually separated from one another. In this case, 10 and 2. And here, 9 and 3 are next to one another. After incubation, the top of the fragment of the disc has produced a copy of 11, 12, and 1, between 2 and 10, the original line of the cut, duplicating what was originally present. Similarly, the bottom fragment fills in 10, 11, 12, 1, and 2, between 9 and 3, again the line of the original cut, recreating the whole clock face. So the rule which is followed is that fragments grow and fill in the missing numbers by the shortest possible route. So for any cut, the clock model enables us to predict how the cells will interact and grow to produce duplicated or regenerated pattern structure.